On the quiz that you will take next week at the beginning of lab, there are going to be 5 questions about cells and organelles, things like, name an organelle that's not found in plants or something like that. And there will be 7 questions on this topic, mitosis. Before I even start my video, let me just tell you quite candidly, Amoeba Sisters does a terrific job at explaining mitosis. So a link to the Amoeba Sisters video is right there on your Canvas page so you can watch it. And if you need to learn meiosis as well, they have a video about that. But we soldier on. So mitosis. Mitosis is the name for the group of processes that will happen when a cell gets ready to divide and become 2 cells. Now, there are 2 times when cells divide in living things. This one is the kind of cell division that happens like all the time, every day. The other one that I mentioned, meiosis, that's when cells divide so that they can make an egg or a sperm. And this does not apply to that. We're just talking about if you have lost some skin cells today, they need to be replaced. They get replaced because cells in your skin divide to replace the ones that got lost, right? So mitosis. Mitosis is the name for all the pieces of cell division. Now, let's start off with the concept of interphase. Interphase, let me get my laser pointer. Oops. Let me get my laser pointer going, right? Interphase is this area here. In general, a cell will spend almost all of its life in this period of time interphase. There are some cells, like cells that will become your mature skin cells, they'll never do anything except for interphase, right? So interphase is where cells are living and building proteins and getting bigger. And it's even in interphase that cells will duplicate their DNA. In order to divide, any living cell is like, okay, I'm going to divide, I'm going to become 2 cells, right? Well, each of those cells is going to need a full copy of your DNA. So all of that DNA needs to get copied. That gets done by DNA replication, and we've got a whole lab about DNA and what DNA does. But for this particular class, lab, we're just going to talk about interphases. Yeah. That's what they do when they're alive. And then they'll go into mitosis. And mitosis has got these phases, okay? Now, I put interphase as a stage of mitosis. Technically, is it? No, all right? But in the life of a cell, it starts off with interphase, and then when it divides, prophase, metaphase, anaphase, telophase, and then when it divides, cytokinesis, and then whoop, it goes back to interphase again, all right? So some people put interphase at the end, some people put interphase at the beginning. It doesn't matter because it's a big old circle. You need to know what happens during the different stages of mitosis. You need to know what order the stages are in, and you need to know how to recognize them from a picture. All righty? So let's start with the order. You will see that in Amoeba Sisters video, they have you learn the order as PMAT. PMAT is the order of the different stages: prophase, metaphase, anaphase, telophase. Perfectly good way of remembering them, right? Another way that people remember them is by the mnemonic interesting people meet and talk. So interphase, prophase, metaphase, anaphase, telophase, and I guess cytokinesis is all on its own. We'll talk about the different phases and what they look like in a moment, all right? So first thing you need to know is mitosis is cell division, the type of cell division where you are growing bigger or replacing cells. And there's interphase and then mitosis. And during mitosis, prophase, metaphase, anaphase, telophase. Now, you're probably saying to yourself, Tidal, what if you ask me, what's the second step in mitosis? Is the right answer prophase or metaphase? And the answer is, I won't ask you that, okay? Because you're right, that'd be a tricky question. So don't worry about that. Okay, here we've got interphase. Interphase, that's the cell just living its life, right? And then we have got prophase, where the chromatin starts to condense. And it goes from looking like a bowl of spaghetti to looking like those little X's that we're used to seeing. The truth is, your chromosomes, they only look like X's just during mitosis, right? And prophase is the first step. In prophase, the membrane around the nucleus starts to disappear. Where does it go? I actually don't know that. But the nucleus starts to disappear and the chromosomes turn themselves into these X's. When chromatin goes from bowl of spaghetti to these X's, that is called condensing. Okay, condense. When the chromatin condenses, that happens during prophase, right? Now, yes, it's true that there are many textbooks that divide it up into early and late. We don't. So the next thing you have to recognize is metaphase. And metaphase, all of the chromosomes line up in the middle, So here they all are right there in the middle. Now, how are they organized in the middle? Well, particularly with animal cells, there are structures called centrioles that are depicted right there and right there, okay? There are structures called centrioles that have got like long fibers, and those fibers are called spindle fibers. And those spindle fibers are attached to the central part of every one of those X's. The central part of every one of those X's is called a centromere. That's a lot of centros, right? I find them difficult, so make sure you can keep them straight. The centromere attaches the two sides of the X. By the way, each side of the X is called a sister chromatid, right? When the cells were divided, when the DNA got duplicated, each one of the chromosomes got duplicated. They stay attached in the center, and they're attached to each other by centromeres. And there is a spindle fiber attached to the centromere on this side, and a spindle fiber attached to a centromere on this side. And then the two sides get pulled. And because the two sides are equally strong, when they pull, their equal pulling lines up all of those X's right there in the center of the dividing cell. Right? So the in metaphase down here, all of the chromosomes are going to be lined up in the middle. So M for metaphase, M for middle. Now, what's going to happen next? So all of the chromosomes, the sets of sister chromatids, are going to be lined up in the middle, and they keep getting pulled by those spindle fibers, and the spindle fibers pull and pull and pull and pull, and then finally, poof, they pop apart. And when they pop apart, the two sides of the X's go to opposite ends of the cell, and that is when we are in anaphase. And you can see it looks exactly like that, doesn't it? We've got a bigger picture in a minute. So that's anaphase. Then what is telophase? In telophase, in animal cells, you will see that new nucleuses are being formed, and you will see what's called a cleavage furrow. A cleavage furrow happens when an animal cell is dividing. And it looks like someone has taken this big fat cell and just put like a rubber band across the center of it, and it'll boom, being pinched apart and create two daughter cells. So the mother cell is the cell we start off with. The daughter cells are what we end up with. And then what is sister? The sister chromatids are the copied, that's terrible, you can't read that at all. The sister chromatids are the copied chromosomes that are attached for the time being in the center. Great. All right, interphase. Most of your cells spend most of all of their time in this state. This is where growth happens. This is where the DNA would get copied and sister chromatids would be made. So this is what most of the cells that you look at in any microscope slide will look exactly like this. This is interphase. Is interphase technically part of mitosis? No, not technically. And I won't ask you that technical question, but someday someone might. Technically, it's interphase and then mitosis, so technically. Prophase, so remember PMAT, right? In prophase, now you can actually see the chromosomes. You can see these little funny X's. That's what you can see in prophase. And the nucleus its envelope is disappearing. So let's look at this, okay? Do you see how in interphase you can draw a line around the nucleus, right? You can draw the line right there. You can draw that. That's because it still has a nuclear envelope. But now let's look here. Around all those sister chromatids all attached to each other, you don't see a circle around them. The nuclear envelope has disappeared. So prophase, you recognize two ways. The nuclear envelope disappears. Technically, this is when the spindle apparatus is forming and the chromosomes, they have condensed. All right? So if I ask you what happens during prophase, nuclear envelope disappears, the chromosomes condense, and the spindle fibers form. Spindle apparatus forms. The spindle apparatus is formed by the centrosomes and the centrioles in animal cells, but remember, plant cells don't have centrioles. How do they form their spindle fibers? They just do it without it, because both plant and animal cells have got spindle fibers in both, right? Now, yeah, the centromere is holding the two sides of that X together. And the centriole is only found in animal cells. Individual centrioles form the centrosome, right? Metaphase. In metaphase, all of them are lined up in the center. And look at that. Right there in the center, you can even imagine that this is all spindle fibers, parts of the spindle apparatus. The centrioles would be found in the middle there, and they're pulling on the X's. That lines everything up in the middle, and that's where we are right before anaphase. I'll start the anaphase discussion at the beginning of the next